thing you can always interrupt without any hesitation. Yeah. All right. So like I was saying, I kind of given a brief introduction in the last part of yesterday, the previous class, and what I wanted to talk about was just two paradigms that to conclude what we had done so far. And the two paradigms is like this: that if you're doing flux analysis, then you essentially have some product I, which is under the influence of some enzyme EI, to give you some product PI. Okay, and whereas, and this is for flux analysis. And when you're looking at the other system biology approach that we talked about, in that case, you have some substrate that is feeding in to a node in which you have the EI to give you a product which is EI. Okay, and here we also have a rate here, Ri. Here also you have a rate. This Ri essentially will depend upon normally Si and some parameter set, right? Because E is normally constant there. Here we also have an Ri. Oops. Let me just move this up. Here also we have an Ri, and this is a function of E as it changes with respect to time and set of theta as well as Si. So that's the difference that you see, and this is, let me call it just SV, system biology. Okay, in general. Okay, flux analysis also comes, but it's considered to be slightly different from. So that's something I wanted to just point out to everyone. Now, the other thing that that I wanted you to kind of go through was the three links I'd given on the Moodle, or three search words. One is KEGG, which you've already seen. The other was SBML and SBGN. How many of you have gone back and taken a look at those resources?
So I've taken a look at KEGG. I have to see the others.
Yeah, just take a look at that. You'll find it's quite useful because so much of what we're doing has already, over these years been kind of regularized, or there's a platform that has been created to take care of these things. Initially, it was not kind of reproducible, and there's a lot of work that was done so that things were all on the same platform, and that's what has happened now. Anyway, so let me continue with signal transduction. And to do this, we have, I mean, these are just statistics, when the word was first used in English, archaic, all those things. But interesting thing is, look at this last point here, which I'm showing here, this one. Okay. So this one, what it's telling you is that, that the first time this came in a biological related paper was in Nature in 1975. And this was a transition of light into neural signals that was mediated with the known visual system that that there are some common type of visual pigments and other things. So based on that. And the first time the title appeared with the signal transduction word in it was here. Okay, and that was just about 3-4 years after that. That was also in Nature. And then, there was a widespread use of this term in biology. And now, if you look at it, it'll perhaps be one of the most commonly used terms in biology, currently being used for research. It appears in almost every paper. Okay, and with that, let me just go forward. And this goes to state what I'd said in the previous lecture, that because we have lipid bilayer separating the cell from its environment, we can often ask the question, what came first, the hormones or the receptors? and obviously, just give me. Okay, this phone I can't switch off because anyway, let's see if I can reduce the sound level. The problem is I'll forget and then I'll not hear any other call. So sorry about that. So so the hormones or the receptors came first, and you look at the history of things that happen, and you look at all the data that is there across different species, and you find that the molecules seem to have different kinds of effects on different microorganisms, different kinds of species of microorganisms. So if you have biomolecules that are there in, let's say, humans, you'll find maybe similar biomolecules are also there in other organisms such as algae, sponges, and vertebrates. For example, cacetylamines are found in protozoa. Ephedrine is found in the Chinese herb, and the mating factors are found in the anterior pituitary glands, but it's there also in yeast, and so on. You can go on and on. And what it tells you is that if you were to look at the way these things are behaving across species, it's only that you're having different kind of behavior for the same molecules. For example, serotonin, which controls moods in humans, will actually stimulate spawning in mollusks. Or you can have a prolactin, which controls fat deposition in migratory birds, or the water regulation in newts and salamanders. So obviously these molecules were there, and the cell had to evolve to kind of respond to these, and therefore the receptors came after that. So this is the general agreement, and did I talk about this? Yeah. And there is a history of how all of this study became important, and this whole catalog of events I've shown here with different researchers ends up with something which is very important. That is a story of how adrenaline became an important to be produced. And so I'm not going into details here, but you can see, if you look at the details that I've given here, essentially what would be ethically completely unacceptable in the modern day was actually being carried out about 100 years ago, 150 years ago by researchers who were very famous and were producing in very, very famous or very reputed journals such as the Lancet. So you can see the Lancet is that old. It was there existing so many years ago. Now even when we look at this COVID SARS-CoV-2 spread, we always refer to that first Lancet journal article where it was reported that they had found people not outside Wuhan who were also affected by the virus. So that is taken as a benchmark, and then you go backward from that date to see when was anything earlier that happened or anything later that happened. So anyway, I'll not go into the details of these things. These are just historical. But let's look at the last one. And this is very important because it goes to show how important it is to be very careful about your research and to record everything properly. And this goes to the stage where that Abel and Crawford in 1987, they published a paper where they talked about the purification of substance they call epinephrine. Okay, this is a Greek word, and the corresponding work that was being done in Japan was being done by Jogi Takamine, and he had made an arrangement with the Park Davis Company, and he made a visit to this person's lab, and obviously he was working in the same compound, and he gained advantage by seeing their processes, and then he did not report any or publish any results. He just went straight for the protection by patents. And then he called his compound adrenaline, which is the Latin of that same thing. And then, and obviously there was a slight difference in this and this, because epinephrine as produced by Abel and Crawford had some problems, which was later rectified. But before all of that happened, this person had beaten them to the draw, and he had produced adrenaline and made a huge amount.
So just a question here.
Yeah.
So did he patent adrenaline or did he patent the procedure which he used to manufacture it?
I think both, because that was early enough. So I don't know. I have not looked at the patent really for this one. But obviously the process would have been different also. So he would have got that for that. And since he's naming it differently, I would think that he also paid, because that compound that they produced was slightly different from the pure and correct compound of adrenaline, which is now known to be binding to the receptor. So I think for both, but I'm not certain. I can have to go back and check. Can somebody do it? Maybe somebody can go back and look at this. Somebody can just Google it while we're talking, and we can later on talk about it a little bit more. Right? Okay. So while I'm doing this, somebody can Google.
Yes, sir.
Yeah. So this is the advantage of having. But do you know that again, just to sidetrack a little bit, and this should not be recording. We should not be recording this part in the lecture, but maybe I'll talk about it later after I stop the recording. Just remind me. I wanted to say something. Okay. Anyway, so this is the core slide for this lecture today, and you need to kind of look at this with little attention. So you see, this is the lipid bilayer here, and normally if you have compounds such as oxygen and carbon dioxide, they'll just pass through the membrane. If you have if you have organic compounds, they'll also pass through the membrane. If you have compounds that are small but they are charged and having some kind of a, like an ethanol does have a small chain, 2 carbons, but that also will pass through. But beyond that, you find that polar molecules are going to be blocked, and if they are charged, then they are definitely not going to pass through. So obviously, charge has something to do with what happens inside the cytoplasm, and because of that, but you do need to understand what these signals are. For example, sodium, potassium, calcium, and chlorine would be very important in neuronal cells, right? And there are very specific channels to allow them to come in and go out, especially for sodium and potassium. And so what we see here is that there are these various other methods by which these compounds can go through. And obviously, if you have hormone, if it is a very small molecule and it needs to pass through cell, it can go through. You also have a receptor, and then it has a very specific function when it binds to a receptor. You also have oxygen, small molecules of gases that can diffuse through. Water molecules can pass through aquaporins. You have gated channels. This could be chemical gated or voltage gated. And on either the intracellular or the extracellular side of it, if some chemical compounds bind and alter the structure of this channel, then you can have some other ion that can pass through this. You can have sympoters and antipoters. So the antipoter is that for every molecule which is charged that goes in, some charge leaves the cytosol. And similarly, if you have a sympoters, then there is, of course, it's opposite to that, but you have, if one molecule goes in, it requires another molecule to go in with it to balance the charge, or to overall, ensure that the reactions that are involved are fulfilling. Obviously they've evolved for a certain reaction, and they'll fulfill that requirement by doing this. Okay. So what essentially this cartoon is essentially this here. It's like a boarding pass. It's like a gate. There is a gate there, and unless you show the right molecule, you can't do anything further. So of course, it's not really an exact analogy, but for anything to happen further down, you'll have to pass through with a boarding pass. So that's what is being visualized there. Okay. Now there are these other things where physical transport of materials take place. So I just talked about the channels that control the passage of sodium, potassium, and calcium in nerve cells. And there are other molecules which are also there bound to the membrane, and the glucose PTS system is a well-known example of that, where glucose, mannose, and I think galactose, the three sugars which need to get inside the cell, would only go through if there is they pass through this glucose PTS system, where of course they have a balance of all the ions taking place and the phosphorylation taking place through two cycles of proteins, where charge is made and released, and the currency would be, of course, phosphates in this case. And you also have endocytosis, where this is an internalization process where proteins from one surface go to another via some coated vesicles, and this is a very now it's like common knowledge after we've seen the virus entering human bodies, and you can see various kinds of virus activities have been visualized using electron microscopy, where it is very clearly shown how actually this endocytosis takes place in different stages, stage-wise, how does it progress and enter into a cell, and it's clearly seen that actually the cell membrane merges with the other before it enters. There are other ways of things happening also. For example, once it enters, how does it rupture? There are symmetries, asymmetries on the caspid molecules which form the outer shell of the virus, and so on and so forth. So let me not get there, but let's talk about what's really happening when we look at a signal transmission taking place. So in principle, it's very easy. What it is essentially telling you is that if you have a signal on the outside of the cell, then, and that of course through evolution is known or deemed by the cell, I'm personifying the cell, deemed by the cell to think it's important as for its survival. That means it can have different functions initiated in the cell, or the cell has to respond by initiating different functions inside the cell. Then that receptor and ligand binding will essentially get into what is called a situation where there will be an amplifier enzyme that essentially amplifies the signal. So what kind of amplification can you tell me? Can you give me an example, anybody, of any real life system that you've seen where such an example can be presented that, yes, that this amplification has taken place, or anything like that? Yes? Oh, okay. People want to be silent today. Okay. Yeah. All right. So, well, the one of the examples that you see is, I'll just give you an example to just give you an idea of how this works out. So, an E. coli, which is, let's say, starved for nutrients, is in an environment that is starved for nutrients. Over a period of time, will lose its flagella. Okay, or the flagella motors will lose, and it will start kind of going into a situation where it will become immobile and not move in the system. Then, let's say at that point of time, there is some kind of signal that comes to tell it there is a nutrient in the environment. Let's say a sugar. So the way it responds is that even if there are 1 or 2 molecules of sugar in that environment, it's going to respond. So that's the level of sensitivity it has. The receptor will have to a ligand and to initiate something within the cell. Of course, before anything happens, has to be a gradient, and there is a mechanism by which more signals are required for it to start synthesizing the flagella motors once again, so that it can swim in the direction of the nutrient. So that's a different story altogether, but that's how it works. Similarly, if you have adrenaline in the system, then just 1 or 2 molecules can actually initiate a whole cascade of events and give you the flight or fight kind of response. and it's more easily visible in the wild where you have, let's say, a predator like a lion chasing, let's say, or stalking a prey like a deer or some other uh, wildlife which is there, smaller animal. So very often it just can't even move, whereas in other occasions just run for its life. So that all that happens through adrenaline. Okay. Now let's look at this example here, and in this example, so you see that how the cascade takes place. So every one gives you something further, and every one of those gives you something further, and so in no time this amplification takes place. Now this is also the principle of biosensors that are developed. That means generally any sensor that you have, you'll first have a sensing unit, you'll have an amplification unit, and the transduction unit. So always you can model it by looking at how you model this binding, how you model this amplification, and how you model this signal transduction. So, and of course, the signal transduction, you can go one step further. What is this function? What does it do? And then you can model that. So you can easily break it up into 4 chambers in terms of modeling chambers and then work them out. So here we have shown it as 3. So you have an extracellular binding activity. You have a change in configuration to give you the amplification, or change in configuration of the amplification enzyme. That's the second part. And the amplification itself, that is the third part. Okay. So the best way to understand this are, the GTP binding proteins. So this is one class of proteins which are there, which are very important, and there is a host of cellular functions that are just dependent on this one. Okay. Yes. Any questions so far? Any questions?
Not yet.
It's clear.
Oh, it's clear. Okay. Good. I have to use this one. Not working. I'll see this later. I don't waste my time on this. So let's look at this one now. So this is one of those proteins which everybody is famous. I mean, everybody is a famous proteins that everybody knows how this works, because this is used as a regular protein to discuss this phenomenon that we're talking about. And so you can see there is an extracellular domain, there's a transmembrane domain, and there's a cytosolic domain. And usually they'll all be populating the surface. Now you can imagine that the cell has to respond to so many different chemicals on the outside, and pretty much in that small environment of 1 micron, 0.5 to 1 micron diameter surface, you have embedded on that a host of these receptors to look out for these signals that are coming in from the outside. So that's one thing that you have to keep in mind. So that is a very fluid membrane with movement taking place based on other factors here in the environment. But definitely there is this binding that has to occur between these two when they are close enough. When you have a ligand binding, there's a structural change and there's a pull to bring these things together. And once that interaction takes place, you have what is called a phosphorylation. Phosphorylation, and you can use this word P as power. So essentially it means it gives this tyrosine kinase power to do something. And in this case, it'll provide the docking sites so that something downstream proteins, such as phosphoenol 3 kinase and phospholipase C, can come and bind and then do its job. And because those proteins they cannot get this power, so this power is getting transmitted from here to them. So this is another thing that you're given. This is an autophosphorylation step. So the kinetics will be pretty much like the Michaelis-Menten kinetic. Okay. So if in terms of looking at how this model has taking place, but same way, except that here now you have to discuss how this dimerization takes place, or at least this interaction between these two cytosolic domains take place, so that the next step, which is the docking of signaling proteins which are downstream, come and attach to this to give you some other behavior. Okay, so again, the basic structure is the same. The three layers are the same, but then you have one extra dimension of complexity that arise with this kind of a behavior. Okay, so also look at how, if you were to look at how these different molecules are there present in the milieu of the cellular environment and within the cell, and in higher animals within tissues and outside the tissue. So what you'll see is that that these pathways that are there in biochemical pathways are pretty much well established now. And in biological systems, you can have, for example, in this case, if you're consuming a plant sterol, or you're looking at an animal sterol like cholesterol. So all of these can actually enter this pathway and then give you one of the compounds, which is essentially a steroid that gives you the precursors for testosterone and progesterone, as well as it is also in itself having other anabolic properties. Now, so this is the pathway through which all of these things happen, and as you can see, there is just no way that you could really replicate any of these pathways and these reactions in a chemistry lab without a huge amount of effort. Whereas this through evolution has given you a very easy and elegant way of having things done. So step by step, it takes you through all of those compounds that need to be produced so that you can get this final compound that is required for the cell to take care of other activities. Okay. Now, so the pathways for the synthesis and the pathways of the utilization of these compounds have to be kept in mind, because first of all, these are going to be there. Let's say it's coming in from some source. So either it has to stop from the source, else they have to be degraded by some method. So there are enzymes that are also present, because you can't have the signal continuously on. So there is this degradation that takes place. So keep that in mind. And in yeast, for example, let's look at, now we kind of broaden the structure that we saw in slide 12 right here on my left hand side. I'm showing you. So that slide, if you remember, we had this, or the one before that, 11 and 12, we had this three structure taking place. Now what we're doing is we're kind of generalizing that, and you see that over a range of these signals, there are pheromones, there are starvation signal, high osmolarity, hypotonic shock, or carbon and nitrogen deprivation. That means there's a reduction in certain compounds in the environment. So so all of that is going through one layer. Then you have the next layer where you are looking at the response that is there getting amplified and then brought to giving you a signal. So this is the MAP kinase step that is there, where all of these, and you can see that they have different intermediate compounds that are there, becoming the main compound that is going to be the signal that is arising because of this amplification. And that signal that is arising will actually give you different responses. And of course, the starting compound is different, the ending compound is. Sometimes you can have different starting compounds, or one starting compound give you two different responses. And just keep in mind this structure that I showed you now. And this structure will be revisited when we are talking about motifs within cells, logical motifs within cells. And I'll bring that back in that context, and we'll discuss this in a different context when we talk about that, and we'll look at how networks and logical junctions are created within cells, just based on something like this. Okay. We'll discuss one protein in detail, and this protein is something that most of you already know. So those who are from the maths and textile background, you might have to go and just look at the net, pick up a video, and you'll understand this. This is so well studied that there are many videos, and some of them may be even better explaining than what I'm saying here. But here there is the other added advantage that, of course, we can go back and forth and have question answers. Is anybody here not familiar? I mean, are we having people here who are from other departments also besides the DBB and people who understand, have a good biology background? Is there anybody here who does not understand this? Let's put it the other way around. Or has not seen.
Yes,
Who are these?
yes,
people?
sir.
Nirmal, yeah,
Yes,
okay, you're chemical
sir.
engineering background. Who else? Okay, I'll just explain it anyway. So it seems that you might be knowing. So what I will do is I'll not spend too much of time in that case. So this is the G protein structure. Okay, this is GTPase protein. So this is part of the class of the G proteins which are there, and GTPase is one of them. Now, so this is a heterodimeric protein. Sorry, heteromeric, not dimeric. Yeah, actually, it's more than trimeric also. So I'll show you. Actually, it's yeah. Anyway, let me just discuss this a little bit here. So you can see that the orange chain is the alpha chain. Then you have the beta and the gamma chain, which is blue and green. And so in principle, it's trimeric. But when it's actually acting with when it really starts activating, then it requires to bind to another protein, and we'll talk about that in the next slide. Okay. So it is. So these are the six. So this is a very very central protein in the human sensing system, and also it's part of the reward mechanism that is there in the brain. It's part of the signaling mechanism there, and so drugs and toxins which are there and lead to addiction. Heroin, cocaine, marijuana, all of these lead to addiction. They all actually based on this G protein coupled receptors, and there's a signaling chain that is there. Even and there is this cholera toxin which is there, which essentially attacks the G protein and breaks it down, and so you lose this signaling. And G protein, besides just this part of the signaling, which is shown in the pathway for drugs and toxins, has many other functions also. So if by just blocking this one protein permanently, you can actually essentially kill a person. So anyway, let's look at how this protein works, and from there we'll try to elucidate the basic principle, that is receptor binding, then a modification and amplification of signal, and then the signal action or execution as a function. So let's look at this slide here. So adrenaline is going to bind to its receptor. So that is the binding that takes place in this receptor here. And the GTP protein, which is essentially not transmembrane, it doesn't cross the membrane completely, but it has a membrane binding section domain here. So when this binding takes place, it essentially will have a modification at this end, which allows this domain of G protein to come and bind to it. If it does that, and then this GTP is released, and it can actually have the binding of GTP taking over. So there's a tenfold excess of GTP now, which is present in the cells. So that essentially this will get removed because of the change in structure that takes place, and GTP will come and bind, as shown here. Okay. Now this GTP binding will essentially release these two beta and gamma groups out of this. So this will get released out, but this stays on binding, and to this essentially this part of it, and then this release that is taking, so essentially it will release from there after that, and then once this part is released, it will essentially bind to this adenyl cyclase which is there, and this adenyl cyclase is one of those proteins which will convert GTP to cyclic AMP. And cyclic AMP is one of the most powerful secondary messengers within the system, where it has a huge number of cellular functions depending on where it is present, right, and what it is doing. So for example, in lac operon, this is one of the mediators. So obviously there are many other operons for gene regulation where cyclic AMP plays a role. And besides this, it also can initiate other cellular function in the protein, which can essentially, which will essentially depend upon other proteins getting activated in the cycle. So you can see that this will give you this here, but this binding that is there, because this is present. So as long as this binding is happening, this because of the 10 times excess of this GTP being present in this, is going to churn out a huge amount of cyclic AMP before this stops. Of course, how it will stop and how this whole thing is reversed, there is a whole series of other stages which we will not discuss in this lecture. Sorry. So how do we go about doing this modeling? So let me see if I can erase this out. Oh, I don't know why it's not working here. Should be erased out. I don't know what's happening here. Try one more time and then I'll just ignore it. Okay, so so the modeling that is done here is essentially showing you, first of all, that there's a binding of the ligand to the receptor. So there's a receptor here. The receptor becomes the activated receptor, and that is a part where you see the G protein essentially getting charged and getting reversed back here. Now, the receptor, when it binds, it gives you the LR, and the LR can get activated to LR star, and here also you'll have the cycle taking place. And when some dissociation takes place, this gets released, and then that is, so once this release takes place, this ligand is either broken down or the synthesis of, or yeah, I do not know if there is, it depends. Some receptors are synthesized, so in that case, this will also be removed because the synthesis will stop and gradually this will get removed out from there. So it just depends on the problem itself. So now look at this. Okay. So it will handle this. Okay. So I want you to just pay attention to the fact that actually you'll find that the rates are all given in terms of the concentration that are present within the cell, right? So note that actually these things at the stars, you cannot really always measure them. So usually there'll be a step where they will be removed when you're actually working out the math of this whole thing. So although we have these worked out here, most of the time you'll not be able to find these values. Like I was illustrating in the other example when I was doing this for the MFA, flux analysis for methionine. So I was telling you that you can get for some organisms, whereas for others you can't get. So usually these are difficult steps. This one and this one. These are the intermediates, and they will be difficult to obtain. Also this one. So usually you try to eliminate them and then get the other equations which are independent of these. So that is one of the things to keep in mind as we move forward on this. Okay. See if I can use this to erase. I don't know. Doesn't do it. Okay. I'm going to leave that as it is here. Okay. I need to clean up my board. I think maybe something to do with the way this thing is working here. Oh, anyway. It's going to be bothering us a bit. So anyway, let's look at this other thing that is there. So we all know about apoptosis. How many of you know about apoptosis or don't know about apoptosis? How many of you heard about apoptosis? Nirmal, have you heard about apoptosis?
Programmed cell death, right?
No, sir.
Yes, sir, we heard about it.
Okay. So this is programming cell death. Okay, apoptosis. So programmed cell death, and if you look at this pathway, this is kind of a template for all the ways in which you can have apoptosis take place. You can have signals from the outside as well as internal signals which can actually lead to apoptosis. Both of them are there. So now you see, of course, there are these caspases which are important, of which caspase 9 and caspase 3 are very important. Caspase 3 is not mentioned here, but caspase 8 is given here because of this particular receptor, the facile receptor which is coming in here. And so you can see that there are these whole series of gene regulation that are there, from which one of the molecules which is key to it, BAX, is synthesized, and that can give you this apoptosis. As well as from this one, the Jack-Set method pathway, it essentially can go this way or that way, and it can give you an inflammatory response, or it can give you a signal which will essentially lead to programmed cell death. Now, there are many checks and balances. It's not that because this is predominantly showing you the apoptotic signal pathway, what is not showing you is that there is a balance between this and survival, where it will ask, cell machinery to repair it. So those things are not shown here, but there is also that part of the pathway which is supposed to balance this out. Now, the reason why it's being shown here is because it brings back to our mind, that there are these receptors that are all along the cells. So this is the cell wall, and you can see a huge range of these receptors. The GPCRs, which is essentially the G proteins, class of G protein that we talked about earlier. And you can see adenylate cyclase, that gives you the pKa signal, that goes to the Krebs cycle, goes into gene regulation. And based on where this original came from, you'll have a whole range of other activities that will take place. And if you were to model this, can you guess how many equations will be there? And how many reactions will be there approximately in this pathway? Besides counting, I mean, you can go and elucidate everything. There are other things that are also possible.
About 20 to 30.
Yeah, around the range of 30. And the number of variables will be higher also. So if I were to ask you to do a flux analysis on this, how would you do it? Now, this is a trick question. Yes? Anybody?
Maybe we'll have to write a program for this, because I feel it's way too complicated to be solved by hand.
Yeah. No, it's a trick question because, flux analysis will not work here, because the flux analysis is more for situations where you can talk about time constants which are different, right? Here, everything is nearly in the same time constant. So you can't make those assumptions that you made in flux analysis. And so you'll have to go into the regulation. And there you are going to face a lot of problems, because you have to know the kinetic parameters for each of these steps to do it. So we'll end up actually simplifying this further. But anyway, so that's the way it is. But yes, you're right that we'll have to, once you have come to this level of complexity and this number of variables, you'll have to essentially start modeling it by yourself. Okay. But one other thing you can do is take a look at SBML website and see if they have one of these pathways. Can somebody report that back to me? Who will do that? Will anybody take? Can Ashi try and look at this up?
Yes, sir.
Yes, okay. I think you have an exam the minus this weekend, right? From this weekend
Yes,
onwards.
sir.
So maybe after the minus. So but keep a note somewhere so that we look at this and the SBML website. Okay, somebody can tell me that, or the SBGN.
Yes, sir.
Okay. All right. Okay. Now, giving you another example, and this is again an assignment. So maybe you can do this assignment after. So this will be the second assignment that I'll give to you. What I want you to do is to use these equations and use Simulink, everybody, and then give the results for this. So I'm going to post this as a assignment after your minors, maybe a week or two after that you can submit, not immediately after that. So this will be a second assignment that you'll need to do using Simulink. All right. I'll ask you to do that later on. Okay. So let's stop here for today. I'm not going to go further because the next one is a topic which is an entirely new topic, which is calcium homeostasis. So I'll stop here. If you have any further questions on the topic today, or any other questions related to the previous lecture that I've given, you can ask me. Anything related to the test, you can ask me now.
Sir, I wanted to ask about the schedule of the test. So, when will it be conducted?
No, like I said in the earlier class, this will be a take-home exam, and I will most likely post the assignment on the day of the exam, which is 17th, I'm told. And you are expected to bring it back and submit it by the day before or on the day of the next class, which will happen. And that is most likely going to be on the 22nd, right? So that you'll get the weekend to do the exam. And obviously, honor code and all that applies. I don't know how much you'll be, because I don't know if I'll be able to, I mean, I know how things work in your circles. So all I'm requesting is, that even if you, I mean, I don't know if I can even stop the consultation. Even if you consult, don't copy from each other. Don't write. When you're writing it, when you're doing the work, do it yourself. So, because this is all open book, open notes, open net. So you can also, I mean, if you please do it yourself if you can. If there is something you're deeply stuck, you can ask me. I would prefer that actually you do the work entire. I mean, follow the honor code strictly and do the work yourself. In case there is a consultation that happens, refrain from writing and copying things that your neighbor is doing. Don't copy paste, type in things that you by yourself, and use your own words to give discussions and explanations. So that's the way it is, because there is not much that we can do on this, but I hope because otherwise you won't learn anything. This whole idea is that you get to learn to do something by yourself. So that's my intention. Okay. So shall I conclude for today?
Yes, sir.
Okay.
Yes, sir.
Thank you.
So one more
Thank you,
thing,
sir.
I looked at the patent.
Yeah. Okay. Good. Yeah.
And
sorry.
He patented basically 12 things. So I guess he patented 6 different procedures and like the products from all the procedures as well.
Yeah.
And
good.
like from what I read, that was the first patent based on an enzyme in the US.
Okay. Good. Maybe can you post that on the Moodle, and then people can take a look at it if you just have a link and maybe the first abstract, not even if you put it that thing, right? So then people can see that is there. Yeah. Good. I like that. So actually, more of that should happen, where you are all participating and doing things, and I should be taking the back seat and listening to you. and I can explain things. But if you do things by your own hands, you never forget in your life. That's the important thing. Okay. Thank you and bye, and do well in exams and in the other exams that you're taking.
Thank you, sir.
Okay,
Thank you,
bye.
sir.
Thank you,
Thank
sir.
you, sir.
Bye. Bye.
Thank you.
समझ में आ गया ना क्या क्या सैंपल है